This is a picture of intimate contact in the days of COVID. COVID has changed everything, as Dr. Zapata has already referenced, and it's because of COVID that I'm not in Chile right now, where I want to be. I'm here in my office at NIH, but maybe this is proper because, in a way, this was the room where it happened. And the story I'm going to tell is mostly what happened here at NIH. Now, in the US, we have to give financial disclosures in our talks. And as I've said before, people who work at NIH only dream about having financial disclosures. Wait, just in, NIH has just banned dreaming. So sorry, but I do have other disclosures. First of all, my guiding scientific principle is that to steal from one is plagiarism, but to steal from many is research. Secondly, that nothing I say reflects the position of the US government, which instantly gives it great credibility and relevance. Thirdly, my memory is not as sharp as it used to be, and also my memory is not as sharp as it used to be. When I won the Nobel Prize, Paul Pokros and his wife sent me this card, which said, "There is no elevator to success. You have to take the stairs." And I was very impressed with this message because you do indeed have to take the stairs. And what I want to show you today is the step-by-step progression in the discovery of hepatitis C. And it was not something that we planned. It was not a thought at the beginning that we would discover a new virus or be able to treat a disease to near eradication. It was just a step by step, something that led from one place to the next. Now, hepatitis history actually began with Hippocrates, that Dr. Zapata told us about the Hippocratic Oath. Well, uh, Hippocrates was a great observer, and he was the first to describe icterus, noting yellowness among many of his patients, and also chiros, or hardening of the liver. Now, he attributed this to an imbalance of the four humors, in this case, an excess of yellow bile. Now, while his hypothesis about the four humors did not hold up to peer review, he actually made a profound observation because in his day, disease was considered to be divine intervention, sort of a punishment for poor behavior. But Hippocrates said, no, this is an environmental factor that perturbs the body's humors, in this case, an excess of yellow bile. He didn't know that these environmental factors might be viruses, but indeed, there were many factors, but viruses were one of them. Nothing much happened over the next 2,000 years, except that there were many, many wars. And in every war, many combatants came down with yellow jaundice. One time it was called campaign jaundice, and then it was vaccine-induced jaundice. And ultimately, it was distinguished as two epidemiologic forms, hepatitis A and hepatitis B. But in all these years, there was no observation of the actual viral particle. There was no specific test for any causative agent. And we just went on until early 1960s, when, as Javier indicated, while I was working with Dr. Blumberg, and we were looking at that time for inherited differences in beta lipoproteins, which would show up in an agar gel plate where an antigen diffusing from one well and an antibody diffusing from another well where they met would produce a precipitin line, which generally would stain blue for lipid. But one day I observed a line that did not take up the lipid stain. Instead, it stained red for protein. Turned out this was a line between the serum of a multiply transfused hemophiliac and an Australian Aborigine who just by chance was being tested on that day. But very briefly, this was called the red antigen. And then when it was shown to be associated with the Aborigines in larger numbers, it was named the Australia antigen, I'm sorry, the Australia antigen, and ultimately became the hepatitis B surface antigen. As Dr. Brahm indicated, Dr. Blumberg went on to prove the association between this antigen and the surface protein of the hepatitis B virus. It's a fascinating story that I don't have time to go into today, but Dr. Blumberg did receive the Nobel Prize for carrying this forward from the discovery to its clinical associations. Today, I want to talk about a study we began around 1969 at the NIH. It was a prospective study to determine the causes of transfusion-associated hepatitis, the incidence, severity, and the possible causes. To do this, we followed patients undergoing open heart surgery who were placed on a heart-lung machine. They received a lot of blood because the heart-lung machine required 7 units of blood just to prime it. The key was we then sampled these patients every week to 2 weeks for the first 3 months post-transfusion, and then monthly for another 3 months. Now, initially, we had no test for the viruses to go on, but we measured the serum ALT as a sign of hepatic inflammation, and we set criteria of a certain level of ALT, which we would consider hepatitis if reproducible. The first findings were astounding in that we found that over 30% of patients who were getting multiply transfused developed either icteric or anicteric hepatitis. Most of these were anicteric, asymptomatic infections that we would not have detected unless we were doing this prospective sampling. At that time, John Walsh at NIH did a study showing that if you received at least 1 unit of blood from a paid donor source, commercial blood, you had a 51% chance of developing hepatitis. Whereas if you received only volunteer donor blood, you had only a 7% chance. Thus, in 1970, we were able to switch from a partially paid donor supply to an all volunteer donor supply and to introduce the very first generation tests for the hepatitis B surface antigen. These were not yet commercialized, but we did it just by hand in the laboratory. These two measures done together caused a precipitous drop in the incidence of post-transfusion hepatitis, falling from 33% down to just under 10%, even though the number of units transfused remained the same as shown in the rectangles. There's nothing we've done actually since that time that has been important as changing the source of the donor blood, because we learned in retrospect that these donors were having high-risk behaviors, probably sharing drugs, involved in alcoholism. So it was a dramatic step and the first step in the process of eradicating post-transfusion hepatitis. In 1973, the next step was the development of a more sensitive third generation assay for hepatitis B surface antigen, which allowed us to go back into these cases and somewhat surprisingly found that only 25 to 30% of all the cases were due to the hepatitis B virus, and that there was another non-B entity that was the predominant cause of transfusion-associated hepatitis. Then in 1975, Feinstein, Kapickian, and Purcell, Feinstein showed on your right, using the immune electron microscope, discovered the hepatitis A virus here on the NIH campus. Purcell and Feinstein were our close collaborators. We immediately sent them our non-B cases for hepatitis A testing, and not a single one proved to be hepatitis A related, the only other known virus at the time. It was then, in a brilliant step of deductive reasoning, that we said if these cases were not A and were not B, that we would call them non-A, non-B. And we used that silly term, that non-specific term, because at the time we had not yet proven that this was a transmissible agent. And we didn't know, if it was, how many agents might be involved. And also we thought we would discover the agent in short order, which we did not. So the next step in this progression was to try transmission studies in the chimpanzee model. The chimps did not get sick from these transmission studies. And you can see them here lining up to sign their informed consent. The important point was that we could readily transmit to the chimpanzee, measuring ALT levels only, and were able to transmit infection from patients who had acute or chronic non-A non-B hepatitis, but also from asymptomatic blood donors who had been implicated in hepatitis transmission. So we now knew we had an agent that we were transmitting even from asymptomatic blood donors. The next important step was this patient, Mr. H, now kind of famous because he became the prototype, a non-A, non-B resource. Mr. H liked to blaze trails, and one time while up on a mountaintop, he had a cardiac arrest. He would have died on the mountain, except that his wife was with him, and she gave him CPR and resuscitated him, and eventually he made his way to NIH. His clinical course is shown here on the left, where the blue curve is his ALT elevations. You can see that about 5 weeks post-transfusion, his ALT levels began to rise precipitously and rose to a level of 2,112, at which time he was jaundiced and fairly sick. However, the ALT then subsided relatively rapidly. At point A, we obtained a plasmapheresis unit from this patient and inoculated into a chimpanzee, as shown in the upper right panel, and this caused hepatitis in the chimp. Bob Purcell then took this material and titered it in other chimpanzees and showed it had a chimp titer of 1 × 10⁶.5 chimp infectious doses. At point B, we obtained another pheresis unit, inoculated that into a second chimpanzee in the middle panel, and this did not cause hepatitis. We think it didn't cause hepatitis because if you now go over to the yellow line in the left-hand panel, which is Hcv Rna, which we could measure years later by PCR, you see that right after transfusion at point 0, there was a blip in Hcv Rna at week 1, which was the donor virus coming into the recipient. That seemed then to be controlled, but then another variant came out and began to replicate rapidly, reaching the peak at point A on the yellow line. The titer by PCR was 3 × 10⁷ copies per ml, or almost identical to the chimpanzee titer. And as you can see, then the Hcv Rna fell rather rapidly, so that by point B, when the material was no longer infectious, the titer had fallen by over a log. And by that time also antibody was present, which was complexing the virus, making it less infectious. And the complexes are shown on the bottom panel where we did differential sedimentation. The bottom line is we now had a titered infectious inoculum, the so-called H inoculum, and we had the chimpanzee model. And that led us to move to the next step, which was a study by Steve Feinstone, where he took the H material and did a chloroform extraction and a sham extraction of that material. The sham extracted material caused hepatitis in the chimp, whereas the chloroform extracted material did not, suggesting that the virus had essential lipid in its membrane, which was a key step in narrowing down the possible etiologic agents. In separate studies, we did filtration studies, again, taking filtrates and inoculating them into the chimp. And with these two measures, we were able to deduce that the non-A, non-B agent was small, somewhere between 30 and 60 nanometers in size, that it had essential lipid in its that had a membrane or an envelope with essential lipid. And this narrowed the candidate viruses down to these small RNA viruses, either alpha or flaviviruses. It could have been a hepatitis B-like virus, but we had a lot of evidence that it was not related to hepatitis B. So I felt it was either going to be a new viral class or one of these small RNA viruses. Dan Bradley at the CDC was, I believe, the first one to say that it was most consistent with being a flavivirus, as it later proved to be. And we kept trying to develop a test for this agent, but could not do so. So we switched our attention to the clinical severity of non-A, non-B hepatitis. And working with the liver unit at the NIDDK here at NIH, we biopsied 39 patients initially. And among these, most had very mild to moderate disease, but 10% already had cirrhosis by the time we biopsied them. And 13% had what was then called chronic active hepatitis, which was likely to progress to cirrhosis. Over time, we did repeat biopsies in 20 of these patients. While most of them appeared to be stable and some even seemed to improve, 25% progressed under our observation to cirrhosis. So we wound up with 8 out of 39, or 20% of our patients who developed cirrhosis, a figure which has really held up over the years, somewhere between 20 to 30% developed cirrhosis. And importantly, out of this group, 3 of them died a liver-related death. 3 others had very severe liver disease when they died of their cardiac disease. So we now knew we had a virus that was transmissible, that could cause chronic hepatitis, that could lead to cirrhosis and lead to fatality. Because originally, many people thought that non-A, non-B was just a benign transaminitis. Over the years, while we had shown the clinical severity was evidence, we still had no serologic, enzymatic, radioimmunologic, or even early molecular method to really get a specific non-A, non-B assay or to really further elucidate the nature of the agent. It was then that I wrote this poem of frustration that Javier has alluded to, called I Can't See the Forest for the HBAGs. And I don't have it in front of me, but I think it went, I think that I shall never see this virus called non-A, non-B, a virus I cannot deliver, and yet I know it's in my liver. A virus that we often blame, but which exists alone by name. No DNA, no antigen or DNA, no little test to mark its way. Oh, great liver in the sky, show us where and tell us why. Let us find this elusive virus. Let us find this elusive virus. If we don't publish soon, they're going to fire us. I think this poem had a great influence on Michael Houghton and his group at Chiron, because shortly after that, Chiron cloned the non-A, non-B agent. And for this, Michael Houghton shared the Nobel Prize with myself and Charlie Rice. What he did may seem easy at this point in time, but in 1983, 84, 85, when the studies were going on, this was really groundbreaking work. And what Chiron did was they pelleted a known infectious plasma from a chimp and from a human. They then extracted the RNA. They also did DNA, but the RNA is what proved to be important. With that RNA, they then reverse transcribed it to cDNA, and then importantly, cut the cDNA with restriction enzymes and inserted those into a FOS GT11 expression vector, which had just been found around the early 1980s. This expression vector meant that any genetic information in the original material when infecting E. coli would be expressed by the E. coli into the surrounding medium. So the E. coli were exposed to the FOS GT11 expression vector and then lysed in agar, and the lysates collected on filter paper. Then the third important step was Michael assumed that there would be antibody in patients, even though no antibody had ever been discovered before or described before. And in a painstaking series of experiments where they immunoscreened these clones, the story goes that they screened 6 million clones before a single one was reactive. But once they had a reactive clone, they could subclone it, re-express the sequences, express an antigen, and develop an antibody assay. When they had the antibody assay, the first thing they did was to request the panel. Here's a picture of Michael Houghton on your right when he received the Lasker Award, and Dan Bradley on the left, who was a CDC collaborator that played a big role in these studies. When Chiron sought my panel, I sent it to them. This was a small panel, but every sample in it was either proven to be infectious in a chimpanzee or proven to be non-infectious by coming from a blood donor who had donated 10 or more times and never transmitted hepatitis. And every sample in the panel was there in duplicate. 19 other laboratories had failed to break the code on this panel, but Chiron broke it perfectly. They detected 3 out of 3 chronic non-A non-B carriers in 6 different samples in 6 random locations. They detected it in 2 out of 2 implicated donors in 4 different random positions. They did not detect it in patients with acute non-A non-B because they were measuring antibody and not virus. But later on, these 2 patients seroconverted for antibody. And importantly, they did not detect it in any of 7 well-pedigreed negative controls in 14 different positions. With this information, which was very exciting at the time, around 1987 or 88, we then went into our stored samples, pulled up 15 of our best non-A non-B cases. And in every case, we found that the patient was negative for antibody before transfusion and then positive at an appropriate time after transfusion. We then looked at the blood donors to 25 of these cases, and we found a positive blood donor in 80% by a first generation assay and 88% by a second generation assay. So we could propose that if we introduce this test to blood donor screening, that we might prevent as much as 90% of post-transfusion hepatitis. Well, indeed, that's what happened. If you go to this curve that began with the very high rates before 1970 and then had lowering over time from different interventions. But in 1990, when the first generation test was introduced, our rates were about 4.1%. They then went down to 1.1%. When the second generation test came along, the rates went down further. And by 1997, we no longer could detect any cases of post-transfusion hepatitis. We now have followed people for another decade after this and have not seen any cases of hepatitis B or hepatitis C. This is that same slide. Well, let me just say this. We could estimate from the various incidents over the years and extrapolating that to the US population that there were probably about 4.8 million HCV infections from blood transfusion in the 2 decades between 1970 and 1990. Conversely, we can say that once we introduced HCV testing in 1990, we probably prevented 2.4 million additional transmissions over the next 2 decades. So this is that same slide just showing this downward arrow. And I show this because this is not just a story of post-transfusion hepatitis. This is the story of my life. And as you can see, both post-transfusion hepatitis and my life are on this very steep decline. And I'm just happy that hepatitis seems to be gone and I'm still up here talking about it. I want you to know that over these years, even though I was a government employee and government employees are considered to be quite lazy, that I always gave 100% at work. It's just that it had this peculiar distribution. So today is actually one of my better days in the week. Now I just want to jump from those studies because that was sort of the end of the beginning in terms of transfusion. But then as we continue to follow these patients and we then continue to follow blood donors who are HCV positive, we found that there were kind of 3 general outcomes. That most of the patients with non-A, non-B had this very stable or very slowly progressive course. And that occurred about 60 to 70%. About 20 to 30%, however, had a more severe and rapid progression where they would develop cirrhosis in 15 to 40 years. And a small group seemed to have very rapid progression where they developed cirrhosis in 5 to 10 years. And I think these were people in retrospect who had alcoholic history and maybe coincident NASH. So we got interested in what makes somebody be a slow progressor versus a more rapid progressor. In a study that Patricia Farsi did, we collaborated with, compared the slow progressors in yellow with the rapid progressors in blue. And if you look at the yellow line, you see that the initial rise in Hcv Rna was then curtailed and almost came back to negative. This patient was almost cured spontaneously, but had an escape mutant. This actually was that patient H, had an escape mutant that then took off and then became persistent over time. In contrast, those patients who were rapid progressors never had this early containment, and their immune response was inadequate to lower the viral titer very much. These rapid responders also, rapid progressors also had a very high level of MCP1, which is monocyte chemotactic protein. This is a pro-fibrogenic chemokine that attracts stellate cells and then is produced by stellate cells, resulting in collagen deposition. So there's synergy in these cytokine levels and the subsequent fibrosis progression. In contrast, the slow progressors had higher levels of interferon gamma and MIP1 beta, which were the good cytokines that helped control the infection. I want to mention just a little bit about hepatocellular carcinoma. This was a study we did with Yoshi Tanaka in Japan, Yasu Tanaka in Japan, and Dr. Mizukami in Japan, where we compared genotype 1B in Japan with genotype 1A in the US. And Dr. Mizukami and Tanaka did a molecular clock, a complex mathematical modeling based on changes in sequences over time. And with this molecular clock, you can predict when a given genotype or a given strain diverged from some common ancestor, and then when it spread rapidly in a population. And you can see that in Japan, genotype 1b first diverged around 1880 and then was relatively stable in the population until the 1930s and 1940s. In Japan, during the 30s and 40s, the country was at war, first with China and then during the World War. We now learn in retrospect that the Japanese soldiers were shooting up with amphetamines before battle with a lot of shared needles. And that was the reason for their epidemic, which is now in Japan leveled off. In the US, our divergence occurred in 1910 and then was quiet until 1960s, 70s, and 80s when we had our own drug epidemic, lots of needle sharing. The important point is that there's about a 30 to 40 year difference between these two curves, and that might account for the fact that hepatocellular carcinoma is 8 to 10 times more common in Japan than in the US. And the Japanese have always said, it's just because we have had this virus longer than you, and you'll see that over time your rates will also go up. And they were right. In this nice study by El Sarag, comparing the rates of hepatocellular carcinoma between the late 70s and the late 90s, there's a threefold increase in the US. And that's all due to hepatitis C, as shown on the right. So back in 2009, it was said that assuming no changes in standard of care, the total number of cases of advanced liver disease and carcinoma by 2029 would be fourfold higher than they were in 2009. Well, fortunately, there has been a change in the standard of care, as you well know, and our drug treatments have gotten increasingly good. So we're now at a point with direct acting antivirals where we can cure 95 to 98% of patients with chronic hepatitis C, and even keep cirrhosis in check. Some studies have shown 100% cure rates. So we're into a different ball game now. So it raises the question of whether we can eradicate hepatitis B and hepatitis C. And with hepatitis B, it's a different ball game. We have very effective vaccines already here that have been widely spread throughout the world. We have nucleotide inhibitors, which can suppress viral load to undetectable. So there is a real hope. We want to get to global eradication, but how do we do that with hepatitis C? Well, we just need a miracle. But the miracle has some components. For B, we need universal vaccination. There's a huge global effort to do this in Africa, Asia. We're not there yet, but it's conceivable that we could eradicate B with vaccination. With HCV, we need to detect almost all the carriers through massive global screening. As you know, at most half the carriers have been detected, and I would suspect that in the developing world, much less than half have been detected. We also then would have to have global delivery of these direct-acting antivirals with probably 90% penetration in each population. What I would conceive before we have a vaccine, or if we don't have a vaccine, is that we need a rapid test for HCV that can be done at the field, in the office. and that immediately, if someone is positive, they would be given their first month's supply of medication, or even a whole 3-month supply or 2-month supply. And in one step, you could begin the treatment process. If we could really do this on this massive scale that would be needed, it is conceivable over the next decades that one could eradicate hepatitis C. Obviously, what you really want is a vaccine. The vaccine is getting a boost now. Michael Houghton's group does have an envelope vaccine that has prevented, not prevented disease, but has decreased the incidence of chronic disease in the chimp model. But now that we have these COVID vaccines made by messenger RNA, people are going back to see whether this approach might work for hepatitis C. Overall, these are tough tasks, but I think what we need now, we don't need better tests. We don't need better drugs. We have that. We just need the political, the corporate, the philanthropic, and the moral will to make it happen. And there are efforts in this regard ongoing right now. So those efforts are going to have to be to the young people in the audience. They're not going to come from me, because I have a new philosophy, new perspective on life with age, where I plotted age on the abscissa and give a damn on the ordinate. And this is me right here. I'm fading out of this picture. It's been a great, wonderful life I've had. Been an exciting story. It had so many collaborators, so many people who gotten me here that could not have done without, particularly Bob Purcell, Patricia Farsi, the liver unit at NIH, and then Michael Houghton taking it to the cloning, and Charlie Rice and others developing these great drug therapies. So it is the end of the game for me, or almost end, but I'm happy to have been there, and I'm happy to answer any questions that I can. Thank you.